Good evening. My name is Tagbo Niepa, and I'm a postdoctoral fellow at the University of Pennsylvania, working in the chemical engineering and chemical and biomolecular engineering department. So the topic I'll be discussing today is frontier in genomics and engineering, designing novel electrochemical treatment for infection control. And I chose this topic because it's based on my PhD work, which I think could lead to a very nice translational research program. So how engineers use genetics. To be honest, it's not a long study that we do usually when we are using genetics as an engineer, because we like to understand how we can improve our system most of the time. And genetics is just something that helps us to answer many questions in order to optimize our system. So we always, as engineers, want to identify some problems and be able to respond to these problems by engineering some new technology that could solve them. So what is the problem that we all think of at this point? The problem of infection control. We know that recently it is very challenging for many of the physicians today to cure infection in the clinical setting. And the reason for that is just because of the recurrence of many drug-resistant bacterial infections. So one reason why people always get involved with bacterial infections just because they receive some medical implant. And 50% of the people receiving a medical implant at the hospital end up with a chronic infection. So in the background of my slides, I have a very graphic image that I was trying to hide. Actually, that's the leg of somebody who received a fracture fixation. So that's just a plate of stainless steel that you receive because he had a broken leg. And as we can see, this lead to a chronic infection, some complication just because the plate got infected. So this kind of situation happened to 5% of the people who receive a fracture fixation. And in the US, there are 2 million of cases of fracture fixation, and 5% of them lead to an infection. But the incidence is even higher when it comes to chronic infection linked to heart device-associated infection. So in this case, most of the people who receive a heart implant, 40% among them, we have an infection. So bacteria is beyond this whole story. Most of the time, they will colonize the implant and then lead to complication. In the case of the gentleman that I was referring to, as we can see in the picture in the middle, he got his leg amputated, meaning that bacterial infection can also lead to so many other issues that could even constitute some economic and some social burden to the people who are affected by that. And some of the complications, as I mentioned, would be amputation, but they can also lead to death. According to a 2010 report from the CDC, 100,000 of people die every single year because of nosocomial infection. And the cost associated to the hospitals say is elevated to 45 billion of US dollars. So that's a major issue. Why we can face these issues today? It's just because bacteria keep evolving. So we have a species today that can turn into a monster the next day. And this is what this graph at the end shows here, where we can see very nicely that some bacterial species such as MASA, methicillin resistant Staphylococcus aureus, has become a drug-resistant bacterium in the past 3 decades, and it has been the same also for other species such as Pseudomonas. So why bacteria do resist to antibiotics? Because they have an intrinsic mechanism of resistance to drug. And this is three means. The first one is that the bacterium can produce some type of enzyme that's going to inhibit the activity of those antibiotics used to treat them. The second mechanism, they have a so-called efflux pump. That's a type of cleaning system that the bacteria have, so that when something is introduced in their system, they can expel that using those pumps. The third mechanism is through the genetic makeup. So the bacteria can switch the target of most of these antibiotics so that the antibiotic that killed them yesterday will not kill the next generation tomorrow. But besides this intrinsic mechanism, bacteria can also develop some type of drug resistance just because they can live in a community. So on this cartoon that we can see here, this is an illustration of a medical implant in the body. Once implanted, the implant gets very quickly covered by some bodily fluids, especially blood and its proteins. So these proteins become the receptor of bacteria that will adhere to the implant, and they will start duplicating. And over time, they will start produce what we call the exopolysaccharide matrix, which is just a polymeric material that they secrete in order to protect themselves against the environment. So in this case, we have billions of bacteria living this community system where they can be easily reached by not only the antibiotics, but also by the immune system. So in this case, the bacteria embedded in this biofilm, as we call that, 10 to 1,000 times more resistance to antibiotic compared to the free planktonic counterpart. So in addition to biofilm, there's another thing to keep in mind. Within the biofilm, there's a population of bacteria, a subpopulation of bacteria called persisters population. And the persister cells, they're nothing else than dormant cells. So because they're dormant, most of the antibiotics do not affect them. The antibiotic mostly target the growing cells. So the persister cells are dormant. They will not be affected by the antibiotic because they are not in their full potential, full metabolic potential, so that once the antibiotic drop under a certain concentration, the bacteria can wake up, and then they can repopulate the biofilm that was completely wiped up of all the normal cells, as we can see in this illustration. The last but not the least mean of resistance to many of the antibiotics that we have today is to this extrinsic factor that contribute to the chronic state of an infection. So at the interface of the medical implant and of the film that bacteria generate when they adhere to the implant, you have an increase or a decrease of pH, and this also leads to what we call biocorrosion. So the biocorrosion will lead to the release of so many metal cation or metal ion that will also help feeding the biofilm. And then we're ending up with a very chronic state here. So we're very much interested into this because it is a very challenging problem. And as we mentioned, the statistics show that today there's no real mean to solve the infection control. So today what people use to treat bacterial infection, they rely on surface modification. So they functionalize the surface that will later on go in the body, hoping that we have a delay in terms of attachment of bacterial cells at the interface of this medical implant that will be introduced later. But this has some limitation because it provides only a limited prevention. I mean, it's just a limited prevention system. A second mode is surgery. So people use surgery today to fix bacterial infection. And this, as we know, impede the quality of life of the patient. And this has been proven with our first example on the previous slides. Finally, the antibiotic remain the most preferred mode of curing bacterial infection. And why so? Just because that's the easiest way to target the growing cells, the growing population of cells. But there's a problem with that, the multi-drug resistance. Treating a bacterium with the same antibiotic over and over again can lead to the development of this resistance mechanism where the bacteria switch the target and then become more and more resistant to antibiotic. And for the last 3 decades, the FDA did not approve any new antibiotics, which raised a lot of concern, especially for people who view antibiotic as a mode of eradicating bacterial infection. So we believe that some antimicrobial approach that will work independently of the growth phase of bacterial cells could provide a good potential to eradicate bacterial persistence and bacterial infection. So we came up with this hypothesis after thinking of the work achieved by those leaders. First of them, Joseph Biggers in 1965. He did focus on the bacteriostatic effect of electrochemical current generated to platinum electrode. So in this case, he used platinum electrode to treat E. coli, and finally it led to the discovery of a new compound called cisplatin. Later on, Dr. Rowley focused on the activity, the synergistic activity between the electrochemical treatment and some antibiotics. Later, somebody who focused on his attention mainly on what we call biofilm today is Bill Costleston. And in this case, he focused on the biofilm, and a few years after, in 1992, he started reviving again this interest in using some electrochemical factor in order to kill the dormant population within the biofilm. So inspired by this work, we came up with this hypothesis. Would it be possible to use some electrochemical factor, some low-level electrochemical factor, including electrical current, electrochemical current, and electrochemical potential, to eradicate bacteria that are dormant and that are not responsive to antibiotic treatment? So this motivation will lead us to those 3 points. As an engineer, we always want to think about ways to solve problems. We also want to think about how we can understand the system better in order to improve that and to develop some new technology. So in terms of eradicating, we want to see how we're going to eradicate the bacterial persistent cells using this method. We also want to understand the mechanism beyond the killing using electrochemical factor. And finally, design some type of innovative technology that's going to assist physicians to solve infection control. So the method that we're using is very simple. So we have a culture flask in which we grow the bacteria cells. Then we isolate the persistent cells, which are the dormant population of bacteria cells. We work only with the persistent cell. We treat them with electrochemical current. And finally, we're trying to see how the electrochemical factor affect the viability of the persistent cells. So by doing so, we can very easily collect the persistent cells from any population of bacterial cells. For the cells that we're working with, Pseudomonas aeruginosa, the persistence constitute 1% of the population of the stationary phase of those cells. So when we isolate the cells and we put them in this electrochemical system that we have on the left corner here, this is the result that we get. We're able to kill the cells by about 2 logs, meaning 99%. So the antibiotic alone, tobamycin, in our case here, 1.5 microgram per milliliter, does not kill the planktonic cells. But in the presence of the electrochemical current, the cell's viability is decreased significantly by 2 log. But once we add the tobamycin concentration to this system in which we deliver electrochemical current, we're able to achieve 3 more logs, meaning that we kill the cell by 99.999%. And these digits are very much important because to have an infection, we need at least 1 million of cells per gram of tissue. So if we kill 99.999%, that means we decrease that to a level that is not significant for an infection. So this will perform with the stationary phase cells. We tend to look at also the persistent cell. We tend to look at the stationary phase cells to see if we have the same, we can achieve the same goal, and exactly the same goal is achieved. And we turn our attention to the cells that are in the full potential of growth, so the exponential phase cells. And in this case, again, we're able to kill all of them in only 1 hour using the electrochemical current alone. But when we combine the treatment with the antibiotics, then we're able to kill the cells in 40 minutes instead of an hour. So we call this the electrochemical control of bacterial persistent cells, or again, ECCP. So we achieve that with 70 microampere per centimeter square of direct current generated with a stainless steel electrode. So, and it does not discriminate the growth phase of the cells. It kills them equally. So since it works out very well in the planktonic system, meaning where the cells are in a suspension, we're trying to focus on our intention on the cells that are confined within the biofilm. And by performing the same treatment on bacteria that go on one of the cathode or one of the electrode, for example, the cathode or the anode, we're able to achieve different killing effect. We realize that most of the killing will perform around the cathode, where the treatment only kills the cells, and when antibiotics supply to the treatment again, we have enhanced killing. So since we were able to achieve that with the cathode, we focus our attention to long-term treatment on a cathode going for 1 hour and 2 hours, and we realize that not only that we're able also to kill the total population of cells, but we're mostly able to focus our attention to the cells that do not respond to antibiotic treatment, meaning the persistent cells, and kill them even better. So we tend to understand why we're able to achieve killing with some electrochemical factors, and what of these factors especially is important to achieve such killing, so that if we have to decouple the treatment method, we can know exactly how we can optimize the system. So we first of all start focusing our attention on the electrolytes that are being produced during the treatment. So during the electrochemical treatment using stainless steel have some metal cation that will be released in the treatment. So if we release those metal cation just for 1 hour and we give it back to the cells, this graph shows that the cells do not respond to that. I mean, they're living fine with the same amount of stainless steel released metal cation that is generated even for 1 hour. They can survive under this condition. We decided to focus our attention on another material, carbon electrode, which is a noble material because carbon is not, does not release any of these stainless steel metal cation. So when we apply a treatment using carbon electrode alone, we don't kill the cell by more than 1 log, so meaning 90% of the cell die. And there is no synergy between treatment mediated with carbon electrode and the antibiotic that we supply to the treatment. But when we put back most of the ion that are released from the stainless steel electrode treatment, and then we allow carbon electrode to mediate with DC treatment, then we can achieve some additional killing, about a log of killing. Although there's no synergy again with the antibiotic, but at least we understood that it was very much important for those metal cation to be in the presence of the electrochemical current. So thinking about those metal cation, we wanted to understand which one among them has most of the effect. So if we perform a treatment of using the stainless steel electrode or the graphite electrode, the carbon electrode, we can have many of these ion release, especially with stainless steel electrode. And this has been quantified here using ICP-MS, which is a very nice method to quantify the ions. We see that in 1 hour, we're able to generate about, to release from the electrode about 0.8 micromolar of iron, 0.27 micromolar of chromium, 0.08 micromolar of nickel, and a very negligible amount of manganese. So now we're trying to start understanding which one of these metal cation affect the cells. So we do another system in which we have the salt concentration, the salt of these metals, ions, and then we give it back to the cells to see how the cells do when they're exposed to these different concentration of metal cation. And as we can see on this graph, for iron in the oxidation state of 2 does not affect too much the viability of the cells. Nickel, the same thing. Iron 3 does not affect also at concentration of 300 micromolar, so very high concentration compared to what is released. But when we come to chromium 2 in the oxidation state 2, we can see that after 4 micromolar, we tend to affect the viability of the cell. So this already light on the cytotoxicity of chromium 2 compared to the other ion, which do not affect the cells. So when we go back and then select those specific ion and supplement that to a treatment, an electrochemical treatment, we see that 3 of them seem to enhance the killing activity of the electrochemical treatment, namely nickel 2, chromium 3, and iron 3. So those 3 specific ion contribute greatly in the killing of the cells. So this brought us to this main hypothesis that's going to drive our thinking from now. We know that DC can kill the cells. I mean, electrochemical current kill the cells. But we believe that maybe this is just because by applying a voltage difference across the cells and mediating some electrochemical currents with ion flowing most of the time, we can disrupt the membrane of the cell membrane of the cells and then allow some toxic material to enter the cells. So we thought that maybe we lyse the cells over the 1 hour of treatment, although that's a very low level electrochemical currents that we're generating. But we can lyse the cells and allow many things to go in. And if we're able to see that, we'll be able to understand how the cell die. We'll be able to also to determine which electrode material will be better to perform such treatment in vitro, optimize the system for medical application. So to confirm that, we went and then did a simple experiment. So we used the persistent cells. We gave them, we use all the cells. We treat them with electrochemical current, and then we isolated persistent cells. We realized that we don't have too much persistent cell remaining in the sample, the original sample. This tells us that maybe the DC treatment as a way to wake up the persistent cells. And if this is the case, it's going to be a very nice way to kill the cells, because it's going to be like we're waking them up just to kill them. Because if they don't wake up, they will not be killed by antibiotics. So I think that would be a very nice way to achieve this. To prove that, we went and then selected just the persistent cells, and then we performed a DC treatment using stainless steel or with carbon electrode. And then right after that, we gave them different antibiotic concentration that is not even supposed to kill them. So the first thing that we do, we gave them 1.5 microgram per milliliter of tobamycin, which we know will not kill them at all. Does not even kill the exponential phase cells. By giving that such amount of drug of the cells treated with carbon electrode, we wipe them out completely. We eradicate all the cells, we kill them. Meaning that maybe carbon electrodes, the current generated by carbon electrode as a mean to create some pores in the cells or activate them so that something that was not killing them finally killed them. But we were not able to achieve the same condition with other material, even stainless steel, which we think was very intriguing because stainless steel was very performant. But at least it told us something, that the DC treatment somehow permeates the cells so that toxic material can enter, confirming our hypothesis. To go one step further to confirm our hypothesis, we perform the same treatment using carbon electrode and stainless steel electrode. And then in this case, we allow the cells to be exposed to potassium iodide. So potassium iodide, or PI, is just a fluorescent marker, a fluorescent probe that interact with DNA. So it is impermeable to cells that are alive, that are intact. When the cells does not have any leakage in the cell membrane, PI will never cross the membrane barrier. But after performing such treatment with carbon electrode, we see that we allow more potassium iodide to go inside the cells compared to when the treatment is performed with stainless steel, which means that the DC treatment somehow permeates the cells. So we see 2 electrode materials that we're using here, and both of them have different advantages that we can build upon to design a better treatment. But we went further to see in real time how these look like to the cells that are exposed to such treatment. So for the control, we see that the cells look nice, intact. That's our figure A. But when the cells are treated with carbon electrode, figure B, we see 2 main configurations of cell morphology. The cell membrane is completely lysed. As we can see here, we don't have a very nice intact cell membrane. And also the second morphology was that we saw some type of localization of some dark compartment at each part of the cells. When the cells are treated with stainless steel electrode, which is our treatment and show in figure C here. We see also those specific configuration where the cells is completely lysed, or maybe the cells start producing some thing that we call the OMVs, outer membrane vesicles. And usually when this happens, it means that cells are under certain stress. So this is already some clues for us to follow up with a genetic study. But before that, we want to confirm again that our observation was true. So instead of performing the DC treatment in 1 hour, for 1 hour, as we see here in this condition, we decided to do that by increment. So 20 minutes, 40 minutes, and 60 minutes. So compared to the control that is figure A, figure B shows the treatment with carbon electrode, and figure C shows the treatment with stainless steel electrode. The panel 1 is for 20 minutes, panel 2 is for 40 minutes, and panel 3 is for 60 minutes. And we can see the gradual change that occurred during the treatment. Carbon-treated cells goes by accumulating this type of material inside themselves, and over time that affects even the cell membrane. But as for cell treated with stainless steel, they start producing a lot of proteins, as we can see all this waste being excreted by the cells. So which also gave us some clue about a follow-up study. So to again decouple the activity of those two electrode material, we focus again on the cell treated with carbon electrode. We understood that there was some compartment that were accumulating inside those cells treated with carbon electrode. So we want to see if this is the carbon that goes and stick inside of the cell. So one thing that we perform here in this case is just to quantify all the ions that are present on the cells treated with carbon electrode and the control. And as we can see in this graph, there's no significance in the difference of those different element or chemical element that we're trying to identify, whether for oxygen, carbon, nitrogen. All these chemical elements seem to be to have the same concentration in cell treated with carbon versus untreated cells, which means that most of the things that were accumulated inside the cell was not an external carbon that we're bringing up. So it looked like the treatment caused so much stress to the cells that they reorganize the inside. So we're trying to see this fact to confirm that we did perform some analysis just by quantifying the number of proteins that the cells have on the cell membrane compared to the inside. So in both conditions, the control versus the cell treated with carbon electrode. By doing that, we see a very inverse pattern. So the control cells have more proteins linked to the cell membrane, but inside there are very few proteins, because here we're dealing again with those stationary phase cells that are not very much metabolically active. But the cell treated with carbon electrode here, as we can see, they have more protein inside than protein on the cell membrane, meaning that again, the level of stress that we are applying by performing the DC treatment even change the configuration, the inner configuration of the cells. And it even goes further by making the cells even harder. So we did perform AFM to measure the stiffness of the cell membrane, and we realized that the cells treated with carbon electrode are even stiffer compared to the cells treated with stainless steel or the control cells. On the other end, the cells treated with stainless steel, they were more the membrane potential or the star potential, which is the surface charge of the cells, did decrease. Instead of being greatly negative, it was less negative than it used to be for the control, meaning that the amount of metal cation that we're generating and that goes on the cells allow the cells to have a very low membrane potential. So knowing this, knowing all this information, we meant to do some 2-hour study just to understand really what's going on to the cell level. We know we can see that that's a lot of stress that we are applying to the cell, because they're not the same type of cell that we have when we're dealing only with the persistent cell, the control cells. So we decided to focus on DNA microarray analysis. So that's the only time that an engineers turn to other biologists to ask for help, because we want to understand really what's going on, and if our treatment would not lead to some adverse effect. So we heard that the cell becomes stiffer. Does it mean that the cell acquires some new resistance mechanism? We don't know. We want to investigate all this. That's why we turn toward DNA microarray. So I would not be a very good person to explain really all the details beyond DNA microarray, but there's this nice YouTube video that we can go and see, and that show exactly in 3 minutes the basics around DNA microarray. And if you need more information on the University of Utah website, you can have even an exercise that will take you through the whole process of DNA microanalysis. So you will understand, for example, how I was able to collect the cells, how I was able to isolate the total RNA of the cells, how I was able finally to establish the transcriptional profile of the cells based on the treatment condition and based on the fact that RNA, the total mRNA was isolated. So let's see and take a look of this video.
A microarray is a small silica or glass slide which is the size of a postage stamp that contains thousands or even hundreds of thousands of spots. Each spot contains a synthetic piece of DNA with a known sequence. A microarray consisting of partial sequences from a number of specific genes is created by placing a series of known single-stranded DNA sequences in an array on a microscope slide or a silicon chip. The sequences are placed in a specific pattern so that each position on the slide corresponds to a particular known DNA sequence. The portion of a microarray shown here only contains 6 spots, which are labeled A through F. These spots may correspond to DNA sequences found in 6 different genes. The expression of genes depends on the cell type and the environmental conditions. To study gene expression, messenger RNA or mRNA can be prepared from a particular type of cell, such as a muscle cell, or from cells that have been subjected to an environmental agent, such as a hormone. This mRNA that is expressed from such cells is used to make complementary DNA or cDNA. cDNA is made in a test tube by an enzyme called reverse transcriptase. This enzyme makes a DNA strand complementary to RNA molecules used as a template. Special nucleotides that have a fluorescent probe attached to them are used to make this cDNA. This probe will make the cDNA molecules fluoresce green when exposed to ultraviolet light. Once the fluorescent cDNA has been prepared, it is allowed to hybridize with the DNA samples spotted in the microarray. The cDNAs complementary to the type A mRNAs will hybridize with the DNA spot in the array that contains DNA complementary to the type A cDNA. Likewise, the type D cDNA will only bind to the DNA in the spot labeled D, while the type F cDNA will only bind to the spot labeled F. Since the cells did not express any mRNAs that correspond to the DNAs in the spots labeled B, C, and E, no cDNA is made that will bind to these spots. After hybridization, any unbound cDNAs are washed away, and the slide is examined under a fluorescence microscope, where the spots that contain fluorescent cDNAs can be identified. In an actual microarray analysis, a typical cell would express hundreds or thousands of different mRNAs, so that hundreds or thousands of different spots would be identified. Therefore, microarray analysis is a powerful method to study the expression of many genes simultaneously from a particular type of cell or from cells that are exposed to a specific environmental condition.
Okay, so that's some very nice material showing again the usefulness of this technique with DNA microarray. Today, it's not only DNA microarray that we refer to when we like to understand the transcriptional profile of the cells. We can even we can also use so-called RNA sequencing or RNA-seq to also, that is based on the same principle to quantify how many genes the cells generate when they change behavior. So we saw different response of the cells depending on the electrode treatment that we were performing, and these responses was also expressed by a certain amount of RNA that the cells did produce that we were able to quantify using microarray analysis just to determine, again, what was our physiological condition of the cells, the persistent cells, under our treatment. So, as we mentioned before, the persistent cells, they have a very low metabolic profile just because they are not metabolically active. But then, when we did the DNA microarray analysis, we were very amazed to know that a bunch of genes was highly expressed, depending on if the treatment was performed with carbon electrode or with stainless steel electrode. So this is the graph for the treatment performed just for 20 minutes. So we perform a very short treatment, as we showed previously in this incremental treatment method. Just for 20 minutes, we're able to see that the cells treated with stainless steel electrode was able to express about 250 genes. There were 250 genes that were induced significantly, and 244 genes that were highly repressed. As for the cell treated with carbon electrode in 20 minutes, we had very few genes that were expressed, especially 51 genes that were induced and 32 genes that were repressed. When we go further to perform a treatment for 40 minutes, then the picture becomes something else. We know that a longer treatment mostly affects the cells, just as we saw in our biofilm treatment, where the 2 hours were more efficient compared to the 1 hour treatment. Here, so we compare the 40-minute to the 20-minute treatment, and using stainless steel electrode, we can realize that, again, many genes are involved, 135 genes versus 95 that's claimed to be repressed. But for carbon electrode especially, this is where we see most of the genes involved, 139 genes induced and 47 genes that are repressed. So that's a bunch of genes that were screened out of 5,000 genes, and depending on the data that we got, we saw that many of them have a certain signature that was representative of the treatment method. So here in these slides, what I tend to show the difference between the expression of the transcriptional profile of the cells treated with carbon electrode or stainless steel electrode. So in the carbon electrode treatment, we can see that the cells start producing a lot of proteins, and among most of the proteins that were secreted by the cells was the so-called the pyrosin proteins. So the pyrosin are just like phages that the cells can even produce themselves as a mean of committing some type of suicide, if we can say so. So they're mostly phages that can lyse the cells. So during the carbon treatment, among the proteins that seem to be accumulated inside the cells are those pyrosin related genes. Pseudomonas has 18 of them. In this treatment, there was 17 of those genes that were highly induced, showing that the treatment was affecting mostly the cells under this aspect. Previous study have linked the secretion or the expression of pyrosin related genes as being also a way for the cells to respond whenever they're exposed to some type of ROS, reactive oxygen species. So in this case, we can see that maybe our carbon treatment generates some type of reactive oxygen species that lead to this generation or to the secretion of pyrosin related genes. But beside that, we also saw that the cell somehow tend to activate the resistance mechanism against antibiotics. So many of the genes involved in antibiotic resistance was also upregulated. And this goes along with our previous data showing that during the treatment with carbon electrode, we didn't see any synergy between the carbon treatment and the antibiotic. So whenever we added the antibiotic at the same time, we didn't have very significant killing, but we have to wait until the treatment is performed using carbon electrode. And then when antibiotic was added sequentially, then we can eradicate all the cells. And this is exactly what this data shows here, this microarray, DNA microarray data. Also, the ribosomal biosynthesis was also expressed because the cells, as we mentioned in the carbon treatment, produce so many material inside. But on the other end, the cell treated with stainless steel electrode start producing a lot of things. We saw the proteins, the expression of proteins, secretion and exports. So meaning that the cells start producing a lot of proteins that can even be exported outside of the cells. And this makes us think already of the OMVs, the outer membrane vesicle, that we were able to identify when the cells were treated with stainless steel. And the cells also upregulate function that involve energy. So by trying to pump some material outside. Inside the cells, they accumulate the reactive oxygen species, and which engender what we call the SOS response, when the cell trying to respond or trying to get some relief because of the accumulation of some very toxic elements such as the reactive oxygen species. And finally, the cell treated with stainless steel electrode was very susceptible to antibiotics, and that's what the genetics shows. And this goes along with our previous finding that when DC treatment is performed with stainless steel electrode and antibiotic is supplemented to the treatment, we got a synergistic killing of the bacterial cells. So this is how we use genetics to confirm our vision and to better our understanding. And if we went further using still the genetics, we could see a bunch of genes related to the secretion of OMVs that were all induced under the condition of 20 minutes and 40 minutes. That means the secretion of OMVs start even earlier when the cells start experiencing the accumulation of this waste product inside themselves. Automatically they tend to respond by inducing all this gene and secreting the OMVs, as we saw previously. So we went one step further based on our genetics data, and to go quickly with that, I would say we perform a treatment. We have a list of genes that get involved with the treatment with stainless steel, another list of genes that get involved with this treatment with carbon electrode. We want to see if this expression of genes is really true, can be confirmed. So what we do, we go back, get some mutants of those genes. So we knock out or we delete those functions in the wild type by some genetic transformation. And after that, we put again the cell back to this treatment to see how they respond to this. So without the treatment alone, if we have to go these cells that were the mutant in an LB medium, which is like a growth medium in which we always grow our wild type cells, they go the same way as wild type. So the genetic mutation does not affect too much of the physiology of those cells. When we gave them 1.5 microgram per milliliter of tobamycin while growing the cells, we're able to inhibit those mutants the same way that we do when we gave the antibiotic from the start to the wild type cells. So they have, the wild type and the mutant have the same physiological profile. So knowing that, we want to see what would make the change if this mutation occurred. So by doing this, we wanted to see what would be the concentration of persistent cells in the cell density. And we realized that those mutations have some significant effect on the population of persistent cells. For instance, the last graph here is the graph of the mutant of PA1897, which is a gene with unknown function. By deleting this gene, we see that the level of persistent cells is about 0.01% less than the level of the persistent cell created by the wild type. So this is already very good information for us, showing that if we can create a molecule that would target these specific genes, maybe we can have less resistance to some type of treatment. So Focusing on the genes that get involved in the treatment with carbon electrode versus the treatment with stainless steel, we perform the DC treatment to see how the deletion of those genes affect the viability of the cells, and we found some very interesting response. The mutant of the PA1897 that we just referred to here get very much killed by the electrochemical current generated by the carbon electrode when we only focus on this particular mutant. So not only that the mutants have a very low level of persistent cells, but performing the treatment of the DC treatment using carbon electrode on this specific mutant lead to a high yield of killing, of cell death. We have few genes also that were in both conditions, up-regulated and down-regulated, that also affect variably differently the level of cells. But this information actually tells us very much about how change in the genetic profile can also affect the response to the persistent cells to the electrochemical treatment. So, to conclude, I would say that we did show in a very clear manner that we can perform some treatment if we have to innovate on designing some technology in order to cure infection. This will lead us to understand the makeup of the electrode material and how we can deliver some electrochemical current to cells embedded in biofilm, and then understand what would be the effect of such treatment on the cells. But focusing on two electrode material, carbon electrode versus stainless steel electrode, we get different response. Performing the treatment with 70 microampere per centimeter square of DC affect the cells greatly. Carbon electrode tend to affect the cells by affecting the surface property of the cell, disrupting the membrane function of the cells, and later allowing a sequential antibiotic treatment to completely eradicate the cells. As for the treatment with stainless steel, we're able to understand that if we perform a treatment in combination with antibiotics, we can kill the cells to a higher level. So the genetics here help us to understand how this killing affect the cells, how this killing occur during the treatment. We're able to see a bunch of genes related to biosyn function, related again to antibiotic resistance that were upregulated when the treatment was performed with carbon electrode. On the other side, we're able to see the cell becoming more sensitive to antibiotics just because antibiotic resistance gene was mostly downregulated, and at the same time, export system, the secretion of OMVs was also showing us that the cells were undergoing a lot of stress. So the genetics definitely is something that is at the frontier in bridging us as engineer to scientists, and then can allow us to focus on different aspects of molecular biology to design some innovative way to control bacterial infection. Thank you so much.